Good morning. Thanks for joining us today. My name is Elmar Nurmamedov. I am co-founder and CEO at Celaris Bio. We are a San Diego-based company developing novel technologies for drug discovery, particularly in the field of cellular target engagement. So today I would like to tell you about our new program called Open Target Initiative. Under this program, we will be opening our technology and our new set of reagents for the wide research community. Today, I would like to briefly introduce some key points. We will overview the Open Target Initiative, the OTI program, the program structure, who can participate and who can really benefit from this, the microtag technology workflow and experimental capabilities expected for this program. And I will provide brief examples for case studies and also introduce how you can go ahead and participate. So first off, what is Open Target Initiative? What is OTI? It's an early access program to microtag cell target engagement technology and reagents. It's a patented technology, and now we have come to a point of really allowing the wide research community to access our technology and reagents. The program is designed to really enable hands-on evaluation of microtag technology and reagents. And we also support exploratory studies for protein quantification and cellular target engagement for basic research and also for drug discovery purposes. Participants may select from a pool of 100 plus validated drug targets and or they may propose their new targets for evaluation purposes. What is microtag technology? You may have listened to our previous webinars. Microtag technology is a patented real-time cellular target engagement technology that enables quantitative, deep, advanced mechanistic measurement of drug target interactions in cells using temperature series. Currently, we're enabling four main applications. We can enable discovery of the chemical matter using cellular cell discovery or plated screens. We can do validation of drug candidates after the fact of drug discovery. That's heat validation. Three, we can enable functional cell target engagement to really understand how the cells respond to cell target engagement events. And then finally, real-time imaging. So for the purpose of Open Target Initiative, we are specifically focusing and narrowing down the scope of the application to heat validation. And for the basically, this is the simplest and this is the most straightforward application that we have under our umbrella. So through this Open Target Initiative platform program, we are going to distribute a limited batch of microtag cell target engagement reagents kits to qualifying labs and researchers. Essentially, we will distribute these real-time target kits. We will also respond and facilitate, accommodate 100 plus validated drug targets. There's a limitation, but you can choose up to three or two validated drug targets, depending on the case. We are open to new targets, case by case. And this enables various therapeutic modalities, small molecules mainly, but various type of modalities, inhibitors, allosteric binders, glues, degraders. And this also enables various applications, but specifically, we're trying to stay on the track of heat validation and target profiling. So why are we doing this? Microtag is really designed to tackle challenging drug targets. There are conventional drug targets like easy targets, like well-behaving targets like enzymes with active binding sites. Easy pharmacology because there is a pocket you fit in an inhibitor and then you have a pharmacology. We're specifically tuning for challenging drug targets like membrane proteins, like transcription factors, like other enzymatic challenging targets that do not have defined targetable sites, ligandable sites. They are in the cell in fluid structural state, and therefore you need a completely different cellular biophysical methods to really bring them to pharmacology and really crack them open for drug discovery purposes. So we are specifically looking into the cases of transcription factors. These are very intrinsically disordered proteins. Membrane proteins like GPCRs that sit in large protein structures, in large membrane structures, and other kinds of enzymes that either do not have a specific binding sites or their binding sites that are essentially too conserved among their family members. So we can go through the wide range family of challenging drug targets, but we are seeing some hallmarks of these targets that are really making a call for cellular biophysical technologies. This is why cell target engagement technology exists, and this is where this is the field where microtag cell target engagement is really making a difference. Again, cell target engagement allows us to interrogate these challenging drug targets within the cells because we know that in the cell, these challenging targets undergo various conformational states. There are low energy and high energy states, and depending on their function in the context, in the cellular context, or in a particular functional context, these targets can switch conformational states based on those various context signals. And in the states, they can exist under various, fluid states, transient states, or more permanent states. And then the question is, how do you engage these relevant structural and functional states for drug discovery purposes? And really, microtech cell target engagement technology exists to address these challenges by going through a temperature gradient, by allowing the targets undergo through various transitions, structural transitions. We can capture the ligandable, the discovery states so that we can go ahead and discover the ligands, and we can go ahead and really measure the cellular target engagement in a mechanistic fashion. I will come back to the OTI program, and of course, and I will tell you the specifics of and logistics of the program. So then again, the microtech enables mechanistic cell target engagement. On the left side, you see the conventional cell target engagement. Now, with conventional cell target engagement, you could do a lot with for well-behaved and well-characterized drug targets like enzymes that have predictable behaviors. And the conventional cell target engagement really uses a single point. Usually that is TAG 50, temperature aggregation 50. It's a state at which your 50% of your target is an unwound state, 50% is in a compact state. And by reducing the complex biology of the target into one particular state, we try to understand whether or not it strongly or weakly or how it interacts with your ligands. It works well when it does. It has its limitations. It's dependent on the prior TAC 50 determination. It reduces the complex biology to a single point. It misses important conformational states below TAG and above TAG. And it gives you endpoint measurements. And also sometimes it's prone to false positives, false negatives, like any other technology. Now, on the right side, you see microtech cell target engagement technology. What we do is we measure cell target engagement through temperature series. We do not necessarily bias our measurements to a single point. We don't need to predetermine a TAC 50 point because we will be scanning from any temperature, any low temperature to a defined high temperature. We can go through, let's, for example, TAC 10, 15, 25, 45, 50, and so on, up to the point at which your target is no longer responsible. So we essentially mimic various conformational states that the target might exist inside the cell. This is ideal for challenging drug targets, like we have discussed. It reads through multiple conformational states. It gains a deep mechanistic insight of how your target responds to your ligand engagement binding. It gives you real-time data And it also provides you partial and the total EC50 values of drug target engagement. So really, you can get a lot from a single experiment, single read. How does microtag works? On the left side, you will see that you have to engineer a specific cell, reporter cell for your specific target. Either this is a transiently expressed target or this is a stable expressed target. Depending on the target, depending on the purpose, there are various ways to do that. And currently, we carry more than 100 validated drug targets for transient expression. And exactly this is the list, the repository of targets that we're opening up through this OTI program. Once you have the reporter system, then you will go ahead and you will culture those cells. You will put them in 96 or 3 to 4 well format. You will treat with your drug molecules or various conditions, and you will essentially put that into a real-time qPCR machine. You can program the machine as if you're programming a qPCR reaction, except that it's for real-time cell target engagement purposes. It's a mix and detect system. It can measure transient and stable expressions of the target, and it's also fully programmable for various drug discovery research purposes, as well as for various target families. And that flexibility is really key as we move forward with developing this technology forward. The microtag technology is an enzyme complementation technology. So on the left side, you will see that how the target is built. You have your target, and you will clonally attach a 15 amino acid tag to the N or the C terminus. And this is the microtag or the S tag. And you will put it into your cell of interest transiently or in a stable fashion. You will treat your cells with your drug molecules at various doses. When you are ready to detect the signal, you are adding two reagents, the S protein and the Fred substrate. Now, the S protein is a large subunit. It complements itself with the S tag, creates an active RNase S enzyme. The enzyme will cleave the Fred substrate and will release the fluorescence signal depending on the temperature that you're testing at and depending on the conformational state and the abundance of your target as a result of that temperature challenge. Now, the Fred substrate has a donor and a quencher at its ends. In its uncleaved state, there is no signal. Once it's cleaved with your complemented a S protein, now it releases fluorescence signal in the proportion that your target was complemented, in the proportion your target was essentially abundant and was stabilized by your ligand that you're testing. You could then go ahead and use real-time system for detection of the signal. These are two examples. Beta 2AR, this is a GPCR that has been well characterized, and there are some commercially available ligands that we know that bind to beta 2AR. pH in this case is the ligand that we have measured against beta 2AR in a transient microtag system. We have measured various doses of the ligands starting from, let's say, sub nanomolar range going all the way to close to micromolar range. It's a very strong binder. And what we do is we subject every dose of that ligand with the target to various temperatures. Basically, we scan from a low temperature to any high temperature. What you're seeing here in panel A is a section that goes from 55 degrees to 70 degrees. And you see that at this 55 degree, all of the doses are stacking pretty much on the top of each other. There is no difference. But as you increase to 60 and 65 degrees Celsius, the target opens up. It's conformationally now open. The ligand can go ahead and bind and engage and stabilize. And you will see that now there is more stabilization, more fluorescence coming out of the target, depending on the concentration, on the dose of the ligand. The higher the dose, the more stabilization of the ligand. You can now go ahead and do data analysis either by the slopes of these curves or by the total area under the curve in comparison to your DMSO control. And then you will get the panel B, which is the final data. This is dose-dependent target engagement potency curve. For this particular case, for beta 2 AR pH engagement, you are getting EC50 of 0.6 nanomolar. And we see that this ligand is a stabilizer of your target. We have also done a similar case for PLK1. Now, PLK1 is a polo-like kinase. It's a great drug target, very hot drug target nowadays. And PLK1s, specifically, there are some positive controls that we know come from large pharma companies like this BI 2536. And we have done a similar path. There's, of course, some quality controls we have to do before we carry on with these real-time experiments. But here is the snapshot. We have gone from a lower temperature. In this case, it starts from 40 degrees and can go up to 70 degrees. And we're testing a DMSO, which is our baseline, no treatment. And we can go to a sub-micromolar and even above micromolar doses of this ligand. And you will see that the more we go to the higher we go with the doses of this inhibitor, the more destabilization of the target that we observe. So there is a target destabilization that is induced by this specific ligand, and this has been previously demonstrated by independent scientific work. We can then do data analysis from either the slopes of each individual curves for each individual temperature, or we can do area under the curve analysis, and that will give you the curve on the right side. There is a dose-dependent destabilization with potency around 86 nanomolar. And then again, this has been benchmarked to previous published literature. Now, I'm going to summarize what Microtag does as a technology and then switch to the OTI program. Essentially, Microtag is a new technology that allows you to measure cell target engagement in a real-time fashion. It goes unbiased for target thermal profiling. You do not need to predetermine the TAG50 or otherwise the melting profile of your target because you're going to scan temperatures anyway from low to high temperatures. You can detect both permanent and transient events inside the cell, the various conformational states your target undergoes. It acquires data fast in real time. It can detect both target stabilization, destabilization, and also it's procedurally very flexible, adaptable for various drug discovery or otherwise target profiling type of work. We have validated this technology for various applications. Now we can do target validation, how your target behaves before we can do any drug discovery. And this here is a very research-driven exercise that many labs might be interested in. We have tested allosteric modulators. We have tested active site binders. We have tested molecular glues and degraders. And we have also done heat to lead optimization using this program. Now, for academic purposes, really, all of the above can be valid, but we see that target validation could be direct application together with testing and interrogation of your target with some positive controls or some validated ligands. Microtag integrates well with a real-time qPCR instruments. You might already have some of these machines in your lab already. And this is one of the requirements of Open Target Initiative. At least you want to have one of these machines in your lab. So this would be Thermo Fisher's Quant Studio machine right here, or Azure Systems Cielo real-time system, Bio-Rad's CFX system, or similar systems from Roche and standard BioTools. So any of them would be fine as long as they're programmable for temperature ramping, as long as you can sensitively detect fluorescence signal. So for Open Target Initiative, we have now an atlas of 100 plus validated drug targets. And this is basically a pie chart showing the types of drug targets that we have validated. Now, a big portion of this, the blue portion, is various type of enzymes. Some of them are conventional, well-behaving enzymes. Some of them are challenging enzymes. The pink portion is the nuclear or proteins and or transcription factors that bind to DNA and that do various essential functions inside the nucleus. And this is the most challenging drug target because these are transiently expressed targets and they exist in the cell under various conformational states. We have worked with membrane proteins, that is the gray area. And these would be essentially the three types, the main types of targets that drive this OTI atlas. We also have other structural proteins. We have transporters, we have secreted proteins, and we have RNA splicing proteins. Really, we're not biased to target families. We can onboard new families. But then again, this is the atlas that is today available for research purposes through this ODI program. So just to give you a snapshot of how these targets, 100 plus targets behave in our system. So I have the thermal melting profile of these various targets. So I have centered them around the TAG50. So we can see that there are some membrane proteins that really start melting or they melt just around between 60, 65 degrees Celsius. They happen to be, most of them are membrane proteins. There are some transcription factors right here that happen to melt between 55 to 60 degrees Celsius, case by case. It needs to be empirically determined. And then these lower temperature melters or targets that melt at these temperatures that happen to be enzymatic targets. So we really have scanned, we have a lot of deep data to really guide the OTI experiments, and we're going to open up this for research purposes. So back to the OTI access to microtag reagents. So we are giving away a small batch of microtag real-time target engagement kits. The kit comes with several components. There is a drug target, a transfection-based drug target. So essentially it's a plasmid and it's limited to most cases one or two different drug targets. The Fred substrate, tag binding protein for complementation, assay buffer, and a positive control to essentially check the integrity of the system and to check your machine, to your instrument. And this one kit should be ideally sufficient for 296 well plates. There's a lot here to do initial experiments and to really validate your scientific curiosity and to give us some feedback. What is included? Then again, it repeats some of the previous points, but we will give you access to our validated drug targets. We will provide you guidance and onboarding support. We will provide you guidance and access to microtag workflows depending on what kind of work you would like to do, access to our current recurring target engagement workshops. If you want to share any of your findings with the great scientific community, you're welcome to do that. And then again, this is meant to be a community action where you would ideally show some of this in an educational fashion to your peer researchers. Who is eligible for OTI? OTI is open to academic labs mainly, for nonprofit research institutions and select biotech companies, and we have to evaluate all of these in case-by-case fashion. Applications are reviewed basically on your scientific relevance, the study of relevance, availability of compatible equipment in your lab, and essentially we will run as long as the batch of our reagents will run. So OTI reagents are intended for exploratory experiments, proof of concept studies, and method familiarization. They will not be sufficient to really run drug discovery efforts, but we really want to give you something for you to get familiar with the technology. Program limitations, some of the limitations will be the number of targets. You can ask for more and we'll have to look into it, but typically we're looking at 1 to 2 targets per lab. Participants also kindly agree to use reagents for non-commercial purposes, give us some feedback in 2 to 3 months, and participate in collaboration, data sharing, or otherwise educational series through the workshops. And that is it. So apply for OTI. Here is the QR code. You can scan it or you can enter the link below on our website. Just to summarize, we're going to provide access to our validated 100 plus targets and microtag reagents to qualifying labs. And this will allow you to do various early drug discovery work using this real-time cell target engagement technology. Essentially, reagents, drug target constructs, and onboarding support will be provided. We can always consider new targets of interest if you feel adamant about those. Data remains with the participant, it's your data, and we would appreciate some feedback and some constructive scientific sharing to really enable the field move forward. Thank you so much. I will take your questions. One question, is there a fee for participation for OTI program? This is not a commercial program. We're not trying to sell these reagents, not to you. So for the OTI program, there is going to be basically a nominal fee just to cover the shipment costs. If you are okay with that, that will be pretty much it for academic purposes. For biotech collaborators, there might be an additional participation fee, but then again, these are nominal so that we just go through the logistics and really enable shipping and otherwise operational expenses. Which of the 100 plus targets would you benefit most from real-time target engagement for the OTI purpose? So we have currently these 100 plus targets, and we do not have any bias to which targets you might be preferring. So we will assume that any of these targets are open, and we have tested abundantly some of them. Some of them are tested less abundantly, but essentially all of them are open for this OTI program for the real-time cell target engagement purposes. How do you know that binding at non-physiological temperatures is true ligand binding and not some occult sites that appear when protein starts to denature? Great question. So when we do temperature scanning for cell target engagement purposes, we do basically start from low temperature, say 25, and go to a higher temperature, say 65. And beyond 37, these are not physiological temperatures. So the question is, why do you even bother about these non-physiological temperatures? That's very true. What we're trying to do here is push your target to a point of destabilization. We are trying to unwind the target so that it reveals all of the possible conformational states it may exist under in the cell. Now, some of those conformational states, again, may not be even physiological, but we are really trying to cover a large spectrum of conformational states. So these will be essentially a collection of various conformational states that may not be physiological, but we are trying to elicit a response from your target. At lower conformational states, lower temperatures, your target will respond, but it may not show a response as it binds to the target or as it binds to your ligand. So really, this is a way of really extracting that response from the target as it undergoes through various conformational states. But then again, once we identify the ligands, we want to retest them under physiological conditions and make sure that there is not only just cell target engagement, there's a physiological response of the cell to that ligand. Can you comment on the comparison of tagless CETSA and microtag technology regarding the sensitivity, throughput, cost, etc.? CETSA is a great technology. It stands for cell um, target shift assay. Now, CETSA has opened up the cell target engagement field. It looks into the endogenous cell target engagement. There's no tag here. Essentially, we use one or a pair of antibodies to really look for the abundance of the target as we expose the target through temperature challenge. Now, it works well when it does. It has, in my opinion, a sensitivity issue because your sensitivity is measured through translation through basically band intensity in the Western blot or otherwise your immunoblotting experiment. And sometimes these transient conformational states of your target, even when the ligand is around, may not translate to stabilization or destabilization for you to measure band intensity in an immunoblotting setting. So you might lose some essentially good candidates. There might be some false positives or false negatives. This is why there is a sensitivity issue. In our case, for the microtag, we're not limiting ourselves to, let's say, one temperature, or we're not limiting to immunoblotting bias. We are going to scan multiple temperatures, and microtag enzyme complementation is very sensitive to really give us the signal as it happens in real time. So sensitivity comparison is a big deal. Throughput wise, microtag technology can be used, for early high throughput drug discovery purposes. We have done screens of hundreds compounds, in a plated fashion, or we can even do cellular DEL discovery. Cellular DEL stands essentially for DNA encoded libraries. As you know, a DEL library can include millions of compounds in a single tube. So you don't even need to go through multiple plates. A single tube, millions of compounds. With our technology combined with DELs, we can discover the chemical matter de novo. And that would be the shortcut to a lot of throughput. Cost-wise, I cannot compare because we do not have that information. So for the sets apart, but for the OTI purposes, essentially there is no charge for technology. There's going to be only a nominal participation fee. Are the 100 plus targets listed in one of your slides? We just showed you a representative list of those targets. Not all of them are included. So we can absolutely share the targets with you on a need to basis. So please apply, please send us a message. If your target or anything close to it is in our list, we will go ahead and share that with you. If it's not on the list, we will give you the options of how to introduce that new target into the OTI program. Can we use your plasmid to clone our target of interest if that is not in your list? That's a potential, right? So we can also guide you into the cloning, and we can also, do the cloning on your behalf, depending on the conditions and depending on the user. So there's a good possibility. How does your technology compare to cell target engagement technology by Promega? Great point. Promega's cell target engagement technology, now CETSA and BRETSA, they use enzyme complementation. And that is one technology that comes really close to our microtag cell target engagement technology. there are key differences. The main difference is how the essentially the biochemistry of the assembly. In our case, that is a 15 amino acid microtag. In the case of Promega, that is 12 amino acid small subunit. There are differences in the amino acid composition of these two subunit of these small peptide tags. The Promega tag is heavily hydrophobic, so it might bias your target into essential occlusion, into hiding, and which makes the assay sometimes less sensitive. In our case, it's a hydrophilic tag, and that really allows us to really make the target more sensitive for cell target engagement purposes. That's one point. The second point is the type of the signal that you're receiving from the two systems. Now, in the Promega system, it's a predominantly an endpoint measurement. You go through the procedure, you essentially let the signal develop. It's fast, but then you measure the endpoint signal. And then again, it's not programmatically flexible. In the microtag system, you get the signal in real time, and you can program the real-time qPCR system to really give you the various flexibility for the target type that you're testing, for the target, for the therapeutic modality that you're testing, and for the various cellular conditions. So this real-time application gives you the mechanistic depth into really cell target engagement with your ligand. So it's like PCR versus real-time qPCR type of difference. We can talk more about this, but I just want to limit the comparison to those 2 points. So I think it's the final question here. I don't see any more questions. For the Open Target Initiative program, what would be the first question or first experiment you would want the users to run? Great question. And we will provide you guidance as we onboard the potential users. Please apply. So the first experiment that we want you to run is, does the target respond in your hands in the first place without any ligand engagement? Just go through the real-time experiment, go through temperature series in your machine, in your setting, and then see whether or not your target responds. Ideally, what we want to see is, like in those two cases with PLK1 and beta-2-AR, your target should melt as you increase temperatures, giving you less and less additive fluorescence. If that is provided, great. That is step number one. You can just leave it at that. If you are just doing basic target biology, or if you want to test some ligands, what you can do is now step number two, try the various doses of your ligands now with the target and repeat the experiment, real-time cell target engagement. Now go from, let's say, 0 DMSO, no treatment, and slowly add doses, go from sub-nanomolar to sub-micromolar or above micromolar, and see whether or not you're getting a target stabilization or destabilization. If that is achieved, really the mission is accomplished. And we will be guiding you. We will be providing you some pre-established workflows. So really looking forward to your participation, and hopefully this will end up really well in your hands as well. Thank you so much for joining us. We will see you next time. So also, we will be participating at two conferences next 2 weeks, Drug Discovery Chemistry and AACR conferences. So we'll be at those conferences with multiple posters. So please find us, talk to us, and we'll see you soon. Bye.